Okay, so we're going to get started with a simple practice problem, thinking about our factors that affect flow. So you have a description here of two arterioles, because we're talking about arterioles again today. And you want to tell me which one of these is going to have a greater flow rate. Okay, so hopefully, if you remember our discussions of the physics of flow, and especially our discussion of resistance, this isn't too bad, but let's walk through what we're doing here. Okay, so we're just trying to figure out which of these has greater flow. So we know our equation for flow is the difference in pressure divided by the resistance, right? I'm not making you calculate this numerically, but we're thinking about this as a relationship between these physical properties. Okay. So what we can see for both arteriole A and arteriole B is that our difference in pressure is the same, right? So that delta P on top, we have a delta P of 50 for both of these, right? So delta P plus 50. So we don't have to think about that difference for this example. What we do see as being different between our two arterioles is that internal radius. So here we have an internal radius of 0.025. And here in arteriole B, we have an internal radius of 0.005. Okay. So who wants to remind me of how we relate radius to this overall equation? What property does radius affect? Okay, there.
Resistance.
Yeah. So our radius affects the resistance. And anybody want to tell me what makes the resistance bigger? An increase in radius or a decrease in radius? A decrease in radius creates an increase in resistance. So they're opposites of each other. They're inversely proportional. So all we're looking at here is that arteriole A with its larger radius, right? So it has a greater little r radius. We have an inversely proportional relationship, meaning our resistance goes down. Okay, so if our resistance is lower and our pressure stays the same, we're dividing by a smaller number, so our flow rate is higher. So our answer here would be that arteriole A with that larger internal radius is going to have a greater flow rate. And that's the fundamental setup for basically everything we're talking about today as we start to talk more about variation in our arterioles, and especially in control of blood flow. We're really just looking at how we change these arterioles to change their internal radii, so their diameter is just twice the radius. So we'll see that appearing in the slides as well. So we're going to think about how we do that, why we do that, when we do that. Okay, so questions about this setup. Okay, it really is mostly this simple. Now we're just kind of tacking more stuff onto it. All right. So our arterioles are the vessels that provide most of our resistance in our cardiovascular system. So we have our big arteries leaving the heart. You can see them. When we get into our microcirculation, so stuff we can't really see, we start with our arterioles. And this is where we're going to see a big pressure drop. This is where we're going to see a lot of resistance. And our measure of overall resistance in our system, just as a vocab term, was our TPR, our total peripheral resistance. So most of that is coming from the arterioles as well. Okay. In terms of what they're connected to, after our arterioles, right? So the arteries led into arterioles. After our arterioles, we're going to lead into capillaries, or sometimes meta-arterioles. Those aren't super important topic for us, but we'll touch on them briefly later. But they're basically taking us from where we're pushing blood out of the heart to the capillaries, where we're going to have exchange between our blood and our tissue. Okay. So a property of the arterioles that is very important is that we can change their radius so that we can change their resistance. And the reason we can do this is that they have a layer of smooth muscle in their walls, okay? So the smooth muscle, those rings in the walls of the arterioles, is the reason we're able to contract an arteriole, make its radius smaller, increase the resistance, decrease the flow, or the opposite. We can relax the smooth muscle, so it would expand, the arteriole will dilate, so it has a larger radius, therefore lower resistance, therefore higher flow. So we saw this graph briefly before when we were talking about pressures and pressures in our different blood vessels. So we can see that our arteries have average pressure of 90, 85-ish is the numbers your textbook are giving. Up here at the top of the system, we have a big drop in pressure across the arterioles as we get to the capillaries, which is good because we want relatively low pressure in those capillaries. They have thin walls. We want that blood to kind of slow down so we can have exchange. So this is useful. And then we can see that the pressure in the venules and the veins drops really, really, really low, which is why when we were substituting into our flow equation, we substituted cardiac output for flow. So cardiac output equals our delta P. That difference in pressure was our mean arterial pressure minus our central venous pressure. But then we started ignoring that subtraction of the central venous pressure because the veins are near zero. That's all I'm getting to with that sentence. Okay, so our biggest pressure drop here is across the arterioles. So that's very helpful. Okay. So this is partially because of the large amount of resistance in the arterioles. So as we have a lot of resistance, that slows down the flow. And this is also creating this pressure drop. So you think about on the other side of a really narrow tube, where we have a lot of pressure going in, it has to go through a narrow tube with high resistance. It's going to have a little less pressure on the other end because of that resistance. Okay, so the pressure drop is from about 90-ish to about 40-ish. We do regulate this resistance here, specifically almost entirely in the arterioles, which is why we're focusing. Okay, so if we're looking at an arteriole, right, we have this outer fibrous connective tissue layer, right, maybe a little elasticy, okay? What we're really, really focused on is the smooth muscle here in the arterioles. I would consider that our most important component here of our arterial wall. We also have a little endothelial layer here lining the inside. Okay. So this smooth muscle can contract, can relax, just like our other forms of muscle. It doesn't look exactly like our muscle fibers in our skeletal muscle. It has this these differently shaped sort of teardrop shaped cell bodies. So what smooth muscle looks like. But what we want to think about right here for how it's going to work in terms of constriction and expansion is that when it contracts, it's a ring, right? So think about it as a ring, right? So we're thinking about a ring going around. So when it contracts, that ring gets smaller. So that line with arrows I just drew gets shorter, meaning the ring gets smaller like that. Smaller line. That's the contracted state. So the relaxed state is going to be larger. So if we make that ring line I drew longer, we get a bigger blood vessel. Okay. So just think about squeezing and expanding our ring, right? Because we're still just shortening and lengthening, just the arrangement is different than before. Okay. So the arterial radius is going to depend based on how contracted that smooth muscle is. So if it's more contracted, we got a little skinny arteriole. If it's more relaxed, we got a big, dilated, large radius arteriole. Now, smooth muscle, and specifically smooth muscle in the arterioles, it does have a certain amount of tone. So tone is like, it's in a slightly contracted state to begin with, right? So like most of the time when you're just hanging out, not changing your arterial radius, you're not fully relaxed, you're not at the largest possible state for that arteriole, and neither are you at that most contracted state. So there's like a little bit of tension in that muscle when your body is at rest. Okay. So we have arterial or tone, so a sort of intermediate contraction level of that radius. We are going to have both local controls on that radius as well as extrinsic controls on that radius, but that tone, that initial state, isn't dependent on the extrinsic controls. And it's just kind of where we are. So since we're in this kind of intermediate tone, we then are able to either dilate and expand or to contract and constrict, right? So it's important that we're not at our maximum or our minimum. So vasoconstriction, constricting, squeezing like a boa constrictor, constricts the radius, makes it smaller, decreasing that radius, and therefore upping our resistance. Vasodilation, vaso is for our blood vessels, dilation is that expansion. So vasodilation is relaxation, relaxation and decreased contraction, kind of the same thing, right? Just different way of saying that. Increases our radius, bigger radius, littler resistance, so we can increase our flow in this manner. Okay. The reason we want to do this, one thing, our arterioles are going out to all our different organ systems, carrying that blood from the arteries there. So we're going to want to be able to control how much blood flow is going to our different organ systems. And more specifically, if we're thinking about the cardiovascular system, those organ systems have within them capillary beds. So we're controlling specifically how much blood is going to an individual capillary bed attached to that arteriole. Okay, so that's going to be important because depending on what you're doing, different organs are going to need more or less blood. So greater or smaller percentage of your cardiac output should go to different organs depending on its needs. The other thing we're going to be able to do by regulating the radius of the arterioles is to regulate our mean arterial pressure. So like our overall kind of average blood pressure. We know it's a weighted average from our equations last time, but still kind of an average. We're going to be able to do that because we have this relationship between flow, pressure, and resistance. So as we change resistance from a system-wide perspective, this can also impact the pressure involved. Cool. So we're going to start by thinking about controlling that blood flow to the organs. Okay. So we're going to begin by thinking about intrinsic control. So intrinsic, inside, we're thinking about local factors. So things like happening right next to the arteriole, We're not thinking about nerves, hormones, anything coming from outside. We're thinking about stuff that's just right there where we are. Okay. So some of the intrinsic control of blood flow happens based on what our organs are doing. This is going to change the properties of the extracellular fluid in the tissue of those organs. And that in turn is going to have an effect on the smooth muscle in the arterioles. Okay. So we're trying to regulate the blood flow to the organs based on what they need. The organs and how they work changes the environment around them depending on how hard they're working. There is an effect on the arterioles of how hard these organs are working. So we can do this because we have a typical flow equation, right? So this is just our same flow equation, actually. Right, so flow is equivalent to a difference in pressure divided by resistance. So all we've done here to get this equation, so we're saying, okay, we don't care about all the flow anymore. We care about the organ flow, right? Blood flow to the organ. Okay. Which means we care about the organ's resistance. I'm going to do the bottom first here. So the organ resistance is going to depend on the state of those arterioles that we're about to control with our intrinsic controls, organ resistance. And then our difference in pressure is going to stay the same as it was for our overall equation. We're just going to think here, abbreviate this as our mean arterial pressure, because that's kind of the overall pressure in the system. We could subtract out venous pressure, but it's negligible, so we're not bothering. Okay, so this is just still that same equation. We've just put in what specific properties we're thinking about now. Okay, so we're gonna walk through how we can vary our flow to different organs. Okay. So in this image, we're looking at a very simple schematic of our systemic circuit. And so our heart is pumping out blood through the arteries to all of our different organ systems. Blood goes through the organ, comes back through the veins, returns to the heart. So that AAP would just be like arterial pressure. That VP is just like venous pressure. So that's just telling you that this is the pressure difference. Okay. So in this schematic, we're just seeing the same arterial pressure for all of these organs, the same venous pressure for all of these organs. And we can see that they've drawn tubes that are the same diameter. So is there going to be any variation in flow to each of these organs in this picture? No. Okay. So they have the same difference in pressure here. They have the same radius, therefore the same resistance. So they'd all have equivalent flow in this image. However, that's not reality. Okay. So in reality, our different organ systems will have different blood vessels going to them, different arterioles going to them, and they will have different amounts of flow that they're using. Okay. So this is pretty much the same image, except for they've gone back to using these tubes of fluid instead of showing us an actual heart in a vein. So we're going to think about our pressures here in this column versus our pressures here in this other column. Right, so our difference in pressure, we're going to call the red our arterial pressure. So that's going to be AP. Our pressure over here in our little column is our venous pressure. So VP. So for all of these, we're still going to have the same difference in pressure across them, despite the fact that some are lower down, some are higher up. Because remember, what we care about is the relative difference, not some like absolute level. Okay. Our difference in pressure is still just arterial minus vein. What's varying here should not be surprising, given how we've set it up. We have variation in how wide these tubes are. So in the radius of each of these tubes, A, B, and C, which we're ultimately going to think about as our organs, A, B, and C, Okay. So this means that we have different resistance and therefore different flow. So what do you know about the radius of A relative to the radius of B and C? Bigger or smaller? You can just do thumbs up, thumbs up. It's bigger, right? So A has a bigger radius, right? So it has a bigger radius, which means it's going to have a lower resistance. Oop, that's P, that's an R, okay? So bigger radius, littler resistance, dividing by a smaller number means we have greater flow. And we can see in the chart on the side that that's true. So here for A, we can see that we have a flow of 1.5 liters per minute compared to 1 liter per minute for B, 0.5 liters per minute for C. And our total flow through this system is 3 liters per minute. So we can also calculate percentages, okay? So A, 1.5 liters per minute is half. So this is 50% of our total flow. And these numbers are the type of range you would expect in your cardiovascular system. So here are 3.0 liters per minute. We're thinking of this as our cardiac output. So we're saying here that 1.5 liters per minute going to organ A is taking up half of our cardiac output. As we move down to B and C, we can see that they're decreasing in radius, B slightly larger than C. We can see that that has the expected effect on flow, right? So slightly smaller radius, slightly less flow than A, but more than C, right? For B, so that's leading us to 33% and a rate of a liter per minute. And then C is very little, so we got a little flow rate taking up the smallest percentage of our cardiac output. Questions about this state before we change something about the image? Okay, cool. So this is a more realistic baseline for some of our organ systems. What we're going to do now is think about either an intrinsic or potentially an extrinsic control. We're mostly intrinsic, okay? So say organ B, we don't need as much blood flow here anymore. So our arterioles constrict, they decrease the radius. I want you to take a look at this image now. Take a look at this set of numbers in our little matrix. I'll give you a second, and then we're going to talk about what's happening. So here's how I want you to be looking at it. All right, so the one change we've made in the image is that we've reduced the radius of B, and therefore we've increased its resistance, which means its flow goes down. All right, so that's the straightforward setup, just like in our warm-up problem. So before we had a liter per minute of blood going to organ B. Now we have 0.5 liters per minute going to organ B. So far, so good. Hopefully that part was straightforward. However, what we also notice is that while the flow to A and C, that overall number, has stayed the same, we can see that instead of getting 50% of the flow for A, we're now at 60%. C before had, I believe, 17%. Now it's at 20%, even though its overall number of liters didn't change. And I think that is the potentially slightly confusing part of this image. So I'm going to key you into what's happening there, which is just that increase in radius, or sorry, that decrease in radius and therefore increase in resistance in B did affect our total resistance in the system, right? So in order to get total resistance, so analogous to our TPR, total peripheral resistance, we're basically just adding up the resistance of all of our blood vessels. So we also, by increasing the total resistance, decreased our total flow. So we're no longer looking at divisions of 3 liters per minute. We're now looking at divisions of this lower flow rate, 0.25 liters per minute through the system as a whole. So 1.5 liters is no longer half, it's up to 60%, even though that individual radius, that individual resistance didn't change. And the overall liters didn't change because we have a decreased flow rate through the system. It's just a greater percentage. Okay. Questions about this at this point?
Do they all have the same pressure? The change of pressure on them?
Yeah, for these, we're going with the same pressure for all of them. Yep. Because like, yeah, same pressure. All right. So this is what we're doing in our bodies. We are changing the resistance of the blood vessels in specific organs in order to control how much blood goes to them. So if you're on a run, we know that that takes a lot of oxygen. Therefore, we need blood to carry the oxygen. We thought about that as we thought about metabolism. Right? So you might want to increase blood flow to your skeletal muscle as you're active, as you're exercising. You wouldn't need as much going to your skeletal muscle if you're, for example, sleeping, right? So we are going to be changing resistance in order to appropriately deliver our nutrients and take away waste products. So these are, in part, really locally controlled factors responding to what the tissue surrounding them is doing. Okay. So some local factors are going to control the vascular resistance by changing the radius of the blood vessels, specifically really the arterioles. So they are going to either contract, so vasoconstriction, or dilate, vasodilation, those arterioles. And they're doing that by changing how contracted or relaxed the smooth muscle rings in the walls are. Okay. So we're going to think about the factors that create constriction versus dilation. The main one that we think about, and the one that probably affects you most day to day, are changes in metabolic activity. Okay. So when we're talking about changes in metabolic activity, right? So metabolism, we remember mostly when we're talking about metabolism, we're talking about breaking down glucose in order to produce ATP, in order to have ATP and therefore have energy to create contractions, to do other processes inside our cells, right? So we're producing ATP. What we also know about that metabolic process, right, is that typically if we're doing just normal cellular respiration, we need oxygen because that's our final electron acceptor inside the mitochondria as we're doing that oxidative phosphorylation using the electron transport chain. And a byproduct that we're giving off as we're breaking down our glucose is carbon dioxide. Right? That's why we breathe in oxygen and breathe out carbon dioxide. It's because we're taking in oxygen in order to do metabolism to make ATP, and we're breathing out carbon dioxide. That's all those carbons that we peeled off in the process. Yeah. So this happens at your sort of organism level, but it is also happening in specific tissues at different rates. Okay. We thought a little bit about variations to that as we talked about our different types of muscle fibers. Okay. So what we're going to tell you now is that when we increase metabolic activity, any kind of byproducts that we create that indicate that increase are usually going to cause vasodilation. Because if you think about a tissue that's really metabolically active, that means it's working. It's making more ATP because it needs the ATP for something, right? So in order to continue that, you're going to want more blood flow so that you have more oxygen, so you can keep up that high rate of metabolic activity. So the primary one that we track through is the carbon dioxide, right? So carbon dioxide, because we're peeling off those carbons and giving them off as carbon dioxide as we break down glucose. But other things that would indicate more metabolically active tissue would be things like potassium and hydrogen ions. We haven't gone into the details on that, but it just happens to be true that those are also high in metabolically active tissue. Okay, so if we see lots of this in our tissue or surrounding our tissue, really, that means we're going to want to carry more blood there. So we're going to vasodilate. Vasoconstriction, on the other hand, is going to be a response to seeing that that tissue isn't very active. So the way we would know that tissue isn't very active is if we have a lot of extra oxygen just hanging around, not being used. And if we're not using that oxygen, that means we're probably not doing much cellular respiration, because if we were doing lots of cellular respiration, that oxygen would get pulled into the tissue and used real quick so that we can make ATP. So if we have extra oxygen hanging around, then we are likely going to vasoconstrict. So making that radius smaller, increasing the resistance, decreasing flow to that tissue, right? As we can better use that blood somewhere else. So we're going to run through an example of this specifically, and we're going to look at some figures that show us how this is happening, kind of. So this increased blood flow in response to increases in metabolic activity specifically is known as active hyperemia. So think active because you are actually active, that tissue is active, maybe you're doing an activity. Hyper for a lot. Emia is an ending. Emia is an ending that means blood. So that's why anemia is a blood related kind of condition. So hyperemia, here we got a lot of blood. So increased hyper, blood flow, emia, and then that metabolic activity is the active part, is how I remember that term anyway. Tells you on the tin when it does. Okay. So in active hyperemia, we're increasing how much blood is going to our tissue because the tissue is doing stuff. It's metabolically active. So it's producing lots of waste products and it's sucking up nutrients. Specifically, it's sucking up a lot of oxygen. Okay. So this first description is of our steady state. So this is like our resting state beforehand. So in our steady state, before we change anything about metabolic activity, the oxygen that's going to our tissue is getting used as fast as it's delivered. And our carbon dioxide, as soon as you produce it, we're just taking it away. So everything's nice and even. We're just kind of swapping one for one. Okay. When you increase the metabolic rate, now we need more oxygen to feed that tissue. So we're consuming more oxygen than we're actually delivering, right? And especially we're producing a lot of CO2, we're producing a lot of waste products faster than your blood can take it away, okay? So we are actually going to see that those concentrations of oxygen and carbon dioxide surrounding the tissue are changing because we've changed its metabolic needs. And that is what your arterioles are going to respond to as a form of intrinsic control. Okay. So if you see low oxygen, right, we have low oxygen because we're using it. So we want to get more blood to deliver more oxygen, means we're going to need to dilate those blood vessels. So vasodilation is a response to low oxygen and conversely to high carbon dioxide, right? Because oxygen and carbon dioxide are kind of trading off. If you're using oxygen, you are also producing a lot of CO2. So high CO2 means you've been doing a lot of metabolisms because the CO2 is a byproduct. So you'd want to increase the blood flow there and sweep that away faster, because having a lot of carbon dioxide hanging out and staying in your body is actually kind of toxic too. It's not good. Okay. So our response to this low O2 and this high CO2 is to increase the blood flow, delivering more oxygen and removing more CO2. So we have some nice little pictures for ourselves. Where can we put the controls? Let's put them down there. actually pop them back up. Okay, so what we're looking at here at the top, when it says cells, we're talking about like tissue, the tissue that I've been talking about as being metabolically active or less metabolically active. So the cells are forming our tissue. Okay. So we know that inside our cells, we have intracellular fluid, our cytoplasm, okay, our ICA, we talked about in AMP1. Outside the cells, we have our extracellular fluid, our ECA. Okay. Specifically, this is interstitial fluid between tissues, okay? And this is where we're going to be tracking how much carbon dioxide and how much oxygen we see. Okay. So in these schematics, we're looking at carbon dioxide as our little green dots. So this is our byproduct of metabolism, and our oxygen is the purple dots. So let me start by circling on oxygen. So what we can see happening is that our tissue needs oxygen to produce ATP through metabolism, through cellular respiration. So we can see our tissue is taking in an oxygen. Here we see our tissue taking in another oxygen over here. And our blood is delivering that oxygen at that same rate. So we had 2 arrows going into the tissue. So we were delivering 2 molecules of oxygen from the blood. So this is a depiction of that steady state. When we say that we're consuming it at the same rate that we're delivering it, that's what we're talking about. So nothing about the concentration in this extracellular fluid is going to change because it's moving into the tissue and just getting replaced by the blood. Same thing is happening on the carbon dioxide side, right? So we're giving off our carbon dioxide as a waste product. We got one over here and one over on the other side. And ultimately, they are then going into our capillaries, getting swept away, and then you're going to breathe them out eventually after gas exchange happens in the lungs. So that's the steady state condition. So I'm going to take a look at how much oxygen and how much carbon dioxide is hanging out here in the extracellular fluid right now, because that's what's going to change in our next image. So just kind of visually gauge that proportion of purple dots to green dots. So now we're looking at what happened if we increased the metabolic rate. So kind of top line, hopefully it's relatively clear. We see a lot more green hanging out in that extracellular fluid now, a lot more carbon dioxide. So the colors have stayed the same. So up on the top, our cells are our tissue. This is our extracellular fluid. These are capillaries. Okay. And leading into the capillary was our arteriole. Okay. So we no longer have a 1-for-1 trade-off because we have metabolically active tissue that has greater demands. So it needs to consume more oxygen. It's creating more carbon dioxide. All right, so if we count them up, right, we're using lots of oxygen. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. These little purple dots, those little oxygens are going into our tissue. Okay. Versus we're still just delivering those same 2 molecules of oxygen. Right. So that is not enough. That's not going to work for us long term. We're depleting all the oxygen that we had in the extracellular fluid. Okay. So this is why we are going to need active hyperemia. This is why it's important, is because we want to adjust this system so that we get back to a steady state where we're delivering enough oxygen at that same rate that it's being consumed. We could do the same thing by tracking the carbon dioxides. We're producing more of these little green dots, but we're still only sweeping away 2 of them, right? So we also need to adjust that part of the system. Okay. So what could we do to fix this? What we're going to do, we're just going to increase the blood flow, right? So if we have faster blood, we have more blood. We're going to have more oxygen. and it's moving more blood, so it's also going to take away more carbon dioxide. The way we're going to do this is changing the arteriole going into the capillary bed. Okay, so what we want to do in this state is vasodilate. So these oxygen concentrations and carbon dioxide concentrations, those changes that we just saw, are going to directly, locally, intrinsically affect the smooth muscle. So the smooth muscle can kind of sense that, and it's going to vasodilate, right? So it's going to relax a bit. It's going to expand the blood vessel, expand the arteriole, so that we have more blood going into that capillary bed. So we can see that in this image, right? Click back. So we had, pretty average looking arteriole, just kind of tubey, normal. We just expanded it, right? So we made it wider. We increased its diameter, increased its radius, therefore decreased the resistance, which increases the blood flow. And now we are delivering a lot more oxygen, right? Before we just had 2. Now we can check out all those oxygens we are now delivering. Got lots of it. So we're basically restoring that oxygen that we had before from that steady state initially in that extracellular fluid. And we're making sure that we have enough for the tissue to take up. So we're increasing the blood flow, getting lots of oxygen there, and sweeping out all the extra carbon dioxide that we produced. Okay. So questions about this overall process? So this is what we mean when we say that there is intrinsic control or local control of the arteriolar radius. We're talking about this, or of arteriolar resistance, because it's both, right? Those are directly linked. Now, we could, we'll talk next here. Okay, so we have a similar thing that happens if blood flow gets interrupted to tissue. Okay, so this next thing is called reactive hyperemia. So we still have the hyperemia. We still want more increased emia, blood, so more blood flow, still our goal. But now we have a reaction. So this is reactive hyperemia. So this is no longer exactly about like increased metabolic activity. This is a reaction to what happens if you stop blood flow, essentially. So imagine we have our arteries going into arterioles, going into capillaries, going to some organ. So you have, I don't know, surgery or something, and they clamp down the artery so that they can cut off blood flow and do a little surgery in that area, right? So for a while, the tissue connected to that artery isn't getting blood flow, right? So it's not getting blood flow, but those cells are probably still doing their thing, right? They're probably still a little bit metabolically active to the best degree possible, given that they have no blood flow. So metabolites are building up. So carbon dioxide that they're giving off as they produce ATP, even in the absence of blood, is kind of building up in that extracellular fluid. That's one of the biggest effects we would notice there. But we'd also see oxygen decreasing. So any oxygen that was in that extracellular fluid is getting depleted as that tissue is still trying to work, even though we don't have any blood. Okay. So if that happens, then the surgery is over, right? You unclamp it, you return blood flow to that tissue. You're going to have a reaction. So a reaction to that previous state, that's the reactive part. So as our reaction, as our response to this previous blockage, we've now restored blood flow, but we need to make up for all the stuff that's been happening while that blood flow is blocked. So we're going to increase the blood flow like to a higher degree than it would have been at normally without a blockage, so that we can readjust those concentrations in the extracellular fluid and therefore restore that like normal steady state. Okay, so if we have a blockage to blood flow, it doesn't have to be actually clamping a blood vessel. Another time you'd really see this, right, is clots, right? A blood clot could block a blood could block blood flow. There are lots of things that could block blood flow. Ultimately, this control is still working pretty much the same, actually kind of exactly the same as with our active hyperemia. It's just that the cause of that buildup or that disruption of the steady state is different. So in a blockage, we would still see the same thing happening, right? Except now we have a different reason. So if we say here between images A and B, we cut this off, right? So we clamped it off, no blood flow would now mean actually we wouldn't really get any oxygen or remove any carbon dioxide. So we would see that same change happening in our relative proportions of carbon dioxide and oxygen because the tissue is still doing stuff. So ultimately, when we release this blockage, so we're blocked here, we'd have an effect. So here we're unblocked, we remove it. We're going to have vasodilation for a little bit until we restore these steady state conditions. So they work the same, local intrinsic control based on concentrations of oxygen and carbon dioxide, just for different reasons. Yeah. I got some flow charts. If you prefer flow chart form, I like pictures, but this is just charting through what we've just discussed. So we have changes to either metabolic rate or blood flow overall, depending on whether we're talking about active or reactive hyperemia. It changes the concentration of oxygen and carbon dioxide. We have a response, vasodilation, decreasing resistance, increasing blood flow, therefore readjusting that oxygen and carbon dioxide until we get back to a steady state. And as we get back to that steady state, this is a negative feedback loop. So we're going to turn it off. And once we're back at our appropriate levels of oxygen and carbon dioxide, we can put the blood vessel back to its normal diameter. So we will pick up next time with our extrinsic controls. I believe we'll talk briefly about myogenic responses. That's just kind of a one sentence sort of description. All right, see y'all Friday.